Hello and welcome back to Ryan Hennessy Wastewater YouTube channel. I am here with some exciting news. I just wanted to make a quick video about the new Wastewater Microbiology book that's available. Spent a lot of work, a lot of time, effort into this, and the goal is really that I hope that it's something that wastewater operators or anyone in the industry can have as kind of a handy reference to keep. And it's purposely made where it's able to very easily get to certain parts for troubleshooting purposes, filament identification strategies, and other things along those lines. So again, very proud of the book. Just a quick overview. Um, there's 10 chapters. The first chapter is an aerobic biological treatment process overview, just kind of talking about the basic parameters in wastewater, the conditions that the bacteria like. Um, Chapter 2, we get into common problems encountered in the treatment process, and we kind of talk about what those different problems are and things along those lines. Chapter 3 is microscopy fundamentals. So it could be applied for, in addition to chapter 3, we're also going to look at putting some content on the channel in terms of, how to press the slide properly and how to align the phase rings, things along those lines. So chapter 3 is more the mechanics of using a microscope. Chapter 4 and 5, chapter 4 is based on low power microscopy, the 10, 20x observations, what we're specifically looking for. Chapter 5, we talk about the higher life form organisms, their importance. Then chapter 6, we're talking about hard SMAS and high power microscopy techniques. So basically, chapter 6 is covering what would be done in a full microscopic evaluation. Chapter 7, we talk about troubleshooting tips and strategies for filament. Chapter 8 would be other problems. So chapter 2 kind of describes the causes of problems, and then chapter 7 and 8 are more specifically targeted towards operational corrections, things along those lines. Chapter 9, we talk about microscopy considerations for other processes like MBR systems, fixed film, lagoons, anaerobic systems. And then chapter 10 covers a study that we did with over 100 samples of wastewater and microscopy and DNA together. And we have some pretty strong correlation between different genus type and different filaments. And we're kind of looking at that list in addition to what's already out there and published. So that said, I'm just kind of quick scan through the book. I'm really happy with the way it turned out. Went with the 12-point font, which I think is a lot more user friendly. And then I did forget to mention at the back of the book, there is a library of all the different recognized filament types that we're looking at under the microscope. So like here, we're looking at traits. So in order to identify filaments, are they, is it true branching, false branching, attached growth, things along those lines. And I'm just going to kind of quickly scroll here to the, towards the back. And then the DNA part, we're just kind of going through, each different filament type and basically what's been proven through fluorescent microscopy applications. And then other things are at this point are just candidates and different theories for different DNA reads that we have that seem to correlate well with filament abundance. And then here at the end is the picture library, and we have all the different types. And the goal is that this could be a nice handy reference for, if you're looking at something under the microscope, you can compare it to, and then it's got the the different characteristics and everything along those lines that we're looking for to identify that. In addition to this picture library in the book, it's also going to be available on the website soon. So again, very proud of this book. I hope that it is useful, and I appreciate any feedback, good or things that can be improved. Always looking to improve things as well. So thank you for your time and have a good one.